Okay, sorry guys. Um, yeah, so I'll just share my screen and we'll begin the lecture. Okay. So as you know, this is one of the practice sessions that is provided to you by NPTEL so that you can, build on concepts that have already been taught to you during your this week's lectures by Professor Laloria. Um, just a moment. I'll just share my screen again. Let me just skip to another kind of sharing. So just as I was saying, this is one of the practice sessions like the ones we've been doing for the past 2 weeks. This is the practice session for week 3. So what we'll be talking about is some of the questions. They could be like model questions or extra questions that we'll solve together in the first segment of this particular session. These will be the previous year's assignment from that were posed for this week three's content, right? Since the week three assessment for this year is currently live, we are not supposed to discuss that and give out the answers, right? But what we can do instead is that we can discuss previous year's assignment. So how that will benefit you is what I have been explaining you time and again. But for people who are new, uh, what like how these questions will benefit you is one, if you are preparing for your exams for this for this particular segment, right? These could be some additional questions that you are prepared for. Apart from that, every question has different kinds of concepts that these questions are built upon, right? And some of these concepts are overlapping also between questions. For example, maybe somebody asks you a question of how does DNA replication occur in prokaryotes, right? So the concept behind that question is going to remain the same, whether that person is going to ask you about the replication for initiation point or the DNA polymerase that is used in that. So we will try to deal with the broader concepts when we solve this question so that other questions linked to that could also be solved, right? So you are basically clear with the concept and the concept has once been revised for you. So having said that, we will begin with the previous year's assignment for week 3. Um, uh, Also, a brief introduction about myself. I am Arveen Kaur. I am one of the TAs for this course, Cell Biology and which is Cellular Organization, Division and Processes. And I am currently a PhD scholar and a PMRF in Department of Biological Sciences and Bioengineering at IIT Kanpur. The materials I will be using for this particular segment of the lecture is material from Professor Shikha Laloria's NPTEL lecture, some material from the book Molecular Cell Biology by Lodish, and some other internet resources and articles that I have cited along the way. Right. So we'll begin with the questions. The first question being DNA replication must occur accurately, right? Why should it occur accurately? To minimize the possibility of spontaneous mutation, to reproduce and maintain the correct genome organization, to ensure the duplication of all parts of the chromosome only once per cycle, or all of the above. So I will give you a moment to think about it. Since we are also recording this and uploading it for your reference later, I will not directly come to the answer. Um, I'll give you a moment to think about it. So the answer for this particular question is all of above, right? I think it is a very simple question. Why? Because we know why DNA replication is occurring. Why do we need DNA replication? Because when cells are dividing, DNA replication basically has to provide more amount of the genetic material that was initially there, so that now it could be sufficient and available for these two new cells that are going to be formed, right? So that makes this B statement true, that is to reproduce and maintain the correct genome organization, right? Also, why should this process be done accurately? Because if it is not done accurately, there could be mutations that could spontaneously arise, right? Mutations such as what if while DNA was getting replicated, there was some deamination in a cytosine base that occurred. Due to this deamination, this cytosine now became a thiamine, right? And if you know the structures of these, you will understand what I'm saying. So if that amine group at one place is lost, it will probably behave like a thymine group now, right? And once it will behave like a thymine group, now when the replication is occurring, instead of G that was supposed to come on the other anti-parallel chain, now what will be targeted? A, because T is complementary to A, right? So this will lead to one base change in that particular DNA that is being replicated. So to minimize such possibilities, we need this particular process of DNA replication to be very, very accurate. And then the final statement says to ensure that duplication of all parts of chromosome occur only once per cell cycle, right? Why do we want this? Because we want exactly the same amount of DNA that was originally there in one cell to be present in different cells at the end of the cell cycle, right? So we want the replication to go on just once, right? Because we are just forming two cells. What if the replication is continuing on, and instead of the two chromosomes that we began with, now we have like 6 copies of those chromosomes? So how will they now be divided between these two cells, right? Instead of diploid at the end, they will be triploid at the end, which is something we don't want, right? It will create some genetic chromosomal alteration, which is known as aneuploidy, which is again uh, not correct, right? So that is the answer for this first question. Moving on to the second question, which says, pick the correct statement about the molecular structure of DNA, right? The options are: the two strands in a DNA double helix run parallel to each other. A binds to T and C binds to G. The base pairs interact with each other by van der Waals interaction, and the four bases present in DNA are A, U, G, and C. So again, this is a very basic, like this question demands a very, very basic understanding of the structure of DNA, right? I hope we are all aware about it. And consequently, in my second segment for today's session, I have laid like a lot of stress upon discussing the structure of DNA in detail. So we'll do that also later. For now, the answer to this particular question is that A binds to T and C binds to G. Now let us see why the other statements aren't correct, right? What does the first statement say? It says that the two strands in DNA double helix run parallel to each other. So is that true? The answer is no. They run anti-parallel to one another. One runs in a 5 to 3 direction, while the other runs in a 3 to 5 direction, right? Then the base pairs interact with each other by van der Waals interaction. That is also untrue, because the base pairs that are A, T, C, and G interact with one another by hydrogen bonding, right? Then the four bases present in DNA are A, U, G, and C. Is that correct? So the answer again is a no, because A, U, G, and C are the four bases that are present in case of RNA, right? In case of DNA, instead of U or uridine, we have uracil, we have thymine, right? So I have also represented this over here that one strand of DNA is running from 3' to 5' direction while the other is in 5' to 3' direction. So they are anti-parallel. Then T is bonding with A and G is bonding with C, which is what we have picked up, right? And the this interaction between this AT and GC is basically hydrogen bond, and apart from that we have already talked that these are the four bases that are present in DNA: A, T, G, and C, right? Okay, so that brings us to question number 3, which says DNA synthesis, pick the incorrect statement about DNA replication, right? So DNA synthesis occurs by extending the 3' end of the primer. DNA polymerase catalyzes the formation of phosphodiester bond. NTPs are utilized as substrate for addition of new nucleotides, and during phosphodiester bond formation, the pyrophosphate is released. So go through these statements very, very cautiously. Um, Although this is another simple question, but at the same time, there is scope to get confused in this one, right? Read the statements once, and we can discuss the answer then. Okay.
Answer C, ma'am.
Answer C is actually the correct one. Uh, yeah. So this is the incorrect statement that is being said about DNA replication, right? Instead of this, what would have been the correct statement? So the correct statement would have been that DNTPs are utilized as substrates for addition of new nucleotides, right? So what is missing over here? The deoxy part, right? Because in case of NTPs, we will say nucleotide triphosphates. If we are not adding deoxy ribonucleotides, then they will probably be confused with um the nucleotides that could be used for RNA synthesis, right, instead of DNA synthesis. So over here, DNTPs should be there instead of NTPs. In that case, this would also have been a correct statement. The other statements are correct, right? Okay. So, question number 4 is something that you will have to recall using your memory regarding last week's lecture. And the question is, the yeast replication origins are called which one of these? ERS, ORI, ATT, or REP? So how you can think
Option
about it is option?
A, ma'am.
Option A,
Yeah,
ma'am.
so yeah. So A is actually the correct answer. How you can remember this is that there is one other replication origin that is present in these options, that is ORI. But ORI is the replication origin in prokaryotes, and yeast is a eukaryote, right? That is the first distinction you need to remember. ARS indeed is the correct answer over here. What is the full form of ARS? It is autonomously replicating sequences, right? And yeast, being a eukaryote, has multiple replication origins that are present. The ARS enables prokaryotic plasmids to replicate inside the yeasts also. And uh, yeah, so this, if you have studied molecular biology uh, in as one of your courses in your colleges, you might remember that this sequence has a very special name that is assigned to it, which is Tata box, right? Why is it called Tata box? Because it has this kind of conserved or consensus sequence, the one that I have mentioned over here, this 11 base pair. Minimal sequence is a characteristic of the ARS, and it is more or less consensus, right? And it is commonly known as the Tata box sequence. Also, additional copies of the above sequence is required for efficiency. That is, this is the minimal sequence. There could be multiple repeats of the sequence that are present, right? Uh, then in case of yeast or S. cerevisiae, which is one um, again the yeast, we can say that there are multiple like multiple components that are present in this origin, right? There are multiple subunits such as A subunit, B subunit, C subunit, and A and B1 in this case form ORC, and the B subunits form another kind of complex, and together these two can form ARS, right? So I think I have yeah. So there are different kinds of ARS again. So what will these kinds of ARS depend on? They will basically depend on what all subunits of A and B type are present in this particular ARS or replication origin, right? So based on that, there are different kinds of ARS also that are present in yeast. Then what is a function of ARS? It is basically one of the replication origins. During the licensing phase in eukaryotes, this replication origin can form this pre-replication complex, right? This can happen at the end of mitosis or in the early G1 phase. And this pre-replication complex is completely formed once this ARS has CDCs and CDTs that are attached to it. Along with it, MCM227 helicase are also attached, right? So now it is prepared to work once it is given a signal that, yeah, helicase start working and start creating that replication fork or the replication loop, it will start working. So this is a pre-replication complex, and ARS has a major part to play in this particular organization. So moving to question number 5, the regulatory point in animal cells, which is crucial for controlling the entry into S phase of the cell cycle, is termed as A, restriction point, B, start, C, entry point, or D, all of the above. Also, an interesting thing to note about some of these questions, the ones that directly are, memory recalling kind of questions that, yeah, so you have read this, you need to recall this. These are the kinds of questions, if any of you are targeting exams like UGC, CSIR, ICMR, right? They have some particular sections in which these direct questions are asked. So you will have like memory-based questions. They could help you in your studies for such exams also. So anyway, for animal cells, which one of these is true? The answer is the restriction point. Okay. If we talk in case of prokaryotic cells, then which one of this would have been true? It would be the start, right? Then start site is present in case of prokaryotes instead of a restriction point that is present in eukaryotes or animal cells. Okay. So this is an illustration of the restriction point. The restriction point could be present anywhere in the G1 in the G1 phase, right? Sometimes in some figures you will see that it is represented in the initiation of G1 when this M to G1 transition is occurring. So there is research that is going on about it. It is not entirely clear to us. But yeah, so what this restriction point is basically is a point at which after which if the cell cycle progresses, then it is committed to go through the entire cell division cycle, right? So it blocks the cell cycle. This point, restriction point, blocks the cell cycle progression unless nutrients and mitogens are continuously present. So, so it is one of the cell's ways of checking whether we have enough supplements, enough nutrients that are present, that if we are sending the cell to go for a division cycle, right? So, so that once it starts dividing or starts replicating, after that we don't face that, oh, this particular substrate is unavailable. Now what will happen, right? So we can't like give up the cell cycle in between. So this particular point is a checkpoint that makes sure of that, right? If a cell does not get through the restriction point, it will not get into the cell cycle. Probably it could go to a G naught or quiescent phase, right? And only when it has proliferation signals and appropriate nutrients that are present will it enter back into the G1 phase and then probably go forth the restriction point as well. Okay. Okay, so I have kept another representation or illustration of the same thing, wherein the timelines of some of these cell cycle phases are also defined, right? G1 being the largest phase, it is a phase where RNA and protein synthesis occurs, but no DNA synthesis happens. Towards the end of G1, there is a restriction point wherein a cell that passes this point is committed to pass into the S phase or the synthesis phase, right? Once the synthesis phase is occurring, DNA synthesis doubles the amount of DNA in the cell, and RNA and proteins are also synthesized during this phase. After which the cell passes to G2 phase, wherein again no DNA synthesis will occur, but RNA and protein synthesis can still continue to occur. After which comes the M phase, which is a short-lived phase, but some of the most important things are happening over here, which include mitosis, the nuclear division, and the cellular division. And the end of mitosis will lead to two daughter cells, right? After this, the cell will decide whether it wants to re-enter the G1 phase or it wants to go to this quiescent and non-dividing phase if the cells, for example, that are terminally differentiated, the final kind of differentiation has been done, right? They will withdraw from the cell cycle and they will enter this resting phase. Some examples of such cells are most of the neural cells, right? Okay. So, question number 6 says, pick out the correct role played by RB in cell cycle, right? So, RB acts as a transcription factor activator, or E2F RB bound complex activates E2F, or RB is phosphorylated by G2M CDK, or active RB inhibits the entry of cells into S phase from the G1 phase. So this one requires a little more recollection than the ones we've come across till now. I will give you a while to go through the question. Try to think about what this RB was. Uh, it uh, was found in a condition known as retinoblastoma, due to which it gets its name. Right? Okay, just give me a. Yeah, so I hope you guys have thought about it. And let's see what the correct answer for this one is, right? So the correct answer for this particular RB is that active retinoblastoma protein inhibits the entry of cells into the S phase from the G1 phase. Right, so let us revise what happens when RB is present and when it is bound to E2F, for example, or when it is phosphorylated or unphosphorylated, right? What happens? So I have picked up this illustration from one of Nature's subsidiaries, that is Oncogene. So it represents a very simplistic cell cycle. As you can see over here, there's G1 phase, S phase, G2 phase, and M phase, right? Um, in this G1 phase, as you can see, there is this RB that we are talking about, this retinoblastoma protein, right? Now, what happens is when retinoblastoma is bound to E2F, right? Now, E2F is a transcription factor. When it is bound to this E2F, it will basically stop this E2F from going and transcribing or initiating transcription, right? But what happens if the correct CDKs that should be present in this particular phase of cell cycle, they come up together. The CDK cycling complex comes up and they form a correct complex. They come over here, and what will the CDK do? It is a kinase. It will phosphorylate its substrates, right? So one of its substrates that it phosphorylates is this RB. Once this RB is phosphorylated, what happens is that it becomes inactive, and it has a phosphate tags on it. Right now, this will go for ubiquital for degradation, right? While the E2F now becomes free. Now, once this transcription factor is free, it can go and it can transcribe or perform transcription, and now the this uh, cell can move on to the S phase, right? Because this particular transcription factor is now free. So the same thing that we talked about right now, there is a better and slightly more detailed illustration that I have brought for the same thing, wherein, so what we'll do is we will break down these this particular illustration, and then we'll try to understand. So what is happening? First, just look at this region. Let me just zoom into that region. First, just look at to this region, right, where it says no mitogenic signals. So what does no mitogenic signal means? That the cell does not have the signals to divide, right? Once that is the case, what happens is that, as you can see, this blue colored complex, that is RB, it is going and it is sitting on E2F and DP, right? Along with some other maybe protein molecules or protein players. Over here, one of them is histone deacetylase, right? Once it is actually going and sitting on these transcription factors, it is not going to let it work, right? But on the contrary, when there are mitogenic signals that are received, right, what will happen? This RB that was covering this these transcription factors and not letting the histone, and it had histone deacetylase, right? So it that was also helping this RB in curbing transcription. Now this RB has been phosphorylated, right? Once it has been phosphorylated, what will happen? Transcription can occur. And once transcription will occur, the cell can proceed into the synthesis phase, right? Now, how will this RB be phosphorylated in the first place, right? So what normally happens is that when this cyclin D CDK4 complex is present, right? Now, the cyclin D CDK4 complex is important. Why is it important? Because once this cyclin CDK complex is present, it will tell that, okay, now the cell cycle can go ahead and it can pass on to the synthesis phase, right? So once it is present, the CDK will act upon this RB. Right, or retinoblastoma protein, and with its kinase activity, it will phosphorylate it, right? Along with that, cyclin E-CDK2 can also help perform the same function. And once that happens, and once RB is phosphorylated, it will become inactive. It will not be able to go and capture these transcription factors. And now the cell cycle progression can occur. Now, what can stop this RB from getting phosphorylated, right? So there are these CDKIs that we talked about, that is cyclin dependent kinase inhibitors, right? So if, for example, there is anything that is not correct, if we don't have enough substrate, if we don't have enough signal, if the correct cyclin CDK is not activated there, or there is some no mitogenic signal that is present, right? This cyclin dependent kinase inhibitor or CDKI could get activated. Once CDKI is activated, it will go and bind to the cyclin CDK complex, and it will not let this CDK perform its kinase activity, right? Once the kinase activity is inhibited, RB will not be phosphorylated and it will go and sit on these transcription factors and the cell cycle will not progress, right? So I hope that is clear. Um, I hope this particular part of the cell cycle is clear. We will move on to question number 7 that we have presented over here, which says that the pre-RC or pre-replication complex consists of which one of these? The ORC, the ORC-CDC complex, the ORC and two copies of MCM complex, or CDT1 and MCM complex. So this is something we have talked about in a different context a while ago. I hope you were looking at what illustrations were being presented. If even if you don't recall it from your sessions that you have attended throughout the week, I hope that if you have gone through those illustrations well enough, you will be able to give this answer, right?
Answer C, ma'am.
Yeah, so the correct answer we here is C. That is ORC and two copies of MCM complex will form the pre-replication complex, right? So when did we see this illustration? When we were talking about ARS, right? It was not the this exact illustration that was present, but when we were talking about ARS, we talked about the pre-replication complex and how helicase or binding of MCM227 is also important on this ARS ORC side, so that the licensing or the pre-replication complex assembly is complete, right? So let us discuss that over here again for people who have joined after we discussed the ARS complex, right? So, for example, in both yeasts and metazoans, both of which are eukaryotic organisms, what happens is that what does the pre-replication complex looks like? It has an ORC. Now, over here, ORC is not visible. So, why is it not visible? Because the illustration is such that this MCM2 is sitting on the ORC. Okay, so you can assume that the ORC is being hidden by this MCM complex over here. And when is the formation of this pre-RC complete? When the ORC that is sitting on the DNA recruits one MCM complex, and this MCM complex is basically a helicase, and it either this recruits another MCM complex or another ORC comes and recruits another MCM complex, but there is two MCM complexes that are present that are head-on collided, right? So I hope you remember that representation from ma'am's lectures, because this is not correctly showing it. But what is happening over here is, what I mean to say is, um, let me just try. Huh. So for example, you say that this is one MCM complex wherein this is the head of the MCM complex and this is the tail, right? And there is this another MCM complex, right? Wherein this is the head and this is the tail. Sorry. So what is happening over here is that when they are present in a complex, they are present like this, right? So both the heads are attached over here. Right, so heads are present over here, tail 1 is present over here, tail 2 is present over here, right? So that is how the pre-replication complexes looks like in case of the eukaryotes. Then after this, what happens is, it is relevant to your next question, so we will quickly see this. What happens is that there is DDK and CDK that come and act on this pre-replication complex, and then they recruit the polymerases, right? And then the replication can eventually begin. So I have not given out the answer for the next question, but let's see if you can tell that. So the next question is, activation of MCM helicase subunits is carried out by DDK, which modifies the MCM helicase by which one of these? So is the modification acetylation? or methylation, or phosphorylation, or ubiquitination? Think about it a bit. Um, I will tell you how to assess it even if you don't know the answer.
Options.
Yeah, so indeed, option C is actually the correct option. Uh, now, it could be possible that you have not been taught specifically that DDK is going and phosphorylating the MCM helicase such that it is getting modified in some way, right? That is very much possible. But at the same time, when such a question is asked, what you have to look at is that DDK, if it is causing some kind of some of these kinds of modification, it is going to be an enzyme, right? So we have to consider DDK to be an enzyme. And probably because it is ending with K, it is a kind of kinase, right? That is a fair assumption to have. So that is a very, very valid assumption to have that this might be a kinase enzyme. Now, what do kinases do? What is the main function of a kinase? All kinds of kinases, they either add phosphate groups, right? Mostly they add phosphate groups, or some of them might be involved with dephosphorylation also. Over here, phosphorylation was the only option that was given, right? Phosphatase is actually phosphorylate. Kinase is always phosphorylate, right? So over here, for example, when if we talk about this ARS or autonomously replicating sequence again, as we talked about, this has an ORC, origin replication complex, and MCM, which is the helicase that was being talked of, right? Now you can see that this helicase over here is phosphorylated, right? And there is another this complex that contains polymerase epsilon, right? Which is basically the polymerase complex that will come and that will sit over here and it will start polymerization and adding some DNTPs to the extended chain, right? And over here you can see that the DDK part or the DDK is phosphorylating the MCM, right? So how is it modifying MCM? It is basically adding phosphate tags over here. And CDK, like we said, if it is ending with a K, it is a fair assumption to assume that this is a kinase, right? If it is a kinase, again, it will phosphorylate. So CDK is also phosphorylating some particular subunits. These are SLD2 and SLD3 that are present in this DNA polymerase complex, right? Okay. So coming to question number 9. Question number 9 says, the semi-conservative model of DNA replication was experimentally demonstrated by which one of these duos? Michelson and Stahl in 1958, Mira and Troy in 1960, Charles Augustus and Magnuson in 1957, or Henry Blackwood in 1960. So I think this is a very, very straightforward question with the answer being Michelson and Stahl. I hope you remember what the DNA replication, what the models were that they had hypothesized and how they proved that semi-conservative was the model that was actually true. That is actually how DNA replicates, right? But we are briefly going to talk about it over here. So, for example, this was a very, very good representation that I picked up from Citable. Uh, and this basically shows how Michelson Stahl basically came up with a hypothesis, right? So, what was their hypothesis? That DNA replicates semi-conservatively, right? That was their hypothesis. Then, what is the methodology they followed, and what is the result that they obtained? So the method that they followed was that they took two beakers, right? In one, what they did was they grew bacteria in N15 or N heavy chain medium, right? Sorry, N15 or N heavy medium. Now, what will this do? Once the bacteria are grown in this particular medium, they will basically incorporate in their nitrogenous bases this particular variety of N15, which is a heavy variety of nitrogen, right? And what they did, that they sampled this at 0 minutes, and when they centrifuged the bacteria that had grown in this medium, they received a band at or they received the bands at this heavy chain level only. That is, both the bands had N15 that was incorporated in them, right? So all the parents had this heavy chain band that was present. Then what they did was they transferred some of the bacteria from this first setup into the second beaker that contained N14, which is lighter than this N15, right? And then because the growth media was well supplemented, the bacteria continued to grow, right? After they continued to grow, what these people did was that they took samples after 0 minutes, 20 minutes, that is around one round of replication in case of E. coli, and 40 minutes, that is around two rounds of replication for E. coli. And what they saw was that at 0 minutes, uh, they, although all the parents showed heavy chain, like all of them sent once this CCL based density based centrifugation was done, right? All the parents at 0 minutes showed this heavy chain band only, right? But after 20 minutes, after exactly one round of DNA replication had occurred, what happened was that all the resultant bacteria that were currently present in this medium after exactly about 20 minutes, what was seen that they only had this intermediate band that they gave. The intermediate band meant that one was a heavy chain and one was a light chain, right? One chain had heavy nitrogen incorporated and the other chain had light nitrogen incorporated, right? Then after 40 minutes, what happened was that in the second generation there was half intermediates that were obtained, because now the parent generation was lost, right? uh, because it is continuously growing in a light chain media, right? So half intermediate and half completely new, that were light chain were observed, right? After this, the bacteria, before the bacteria reproduce for the first time in the, oh, this we have discussed, right? Then what were the final results? After two generations, the half the DNA was intermediate and half was light only. There was no heavy only DNA that was now present, right? How can we understand this? Is because the if, for example, we say that there was one parent strand to start with. Now this one parent strand in the first division itself, it got separated, like, right? One of the strand acted as a template for one new strand, the other acted as a template for another new strand, and now these two became separate strands, right? So after the first division was taking place, all these originally heavy ones had gotten separated and acted as templates for new strands to grow on, right? That is why no heavy only strand was observed at the end of this experiment. What was the conclusion of this experiment? That this pattern could only have been observed if each DNA molecule contains template strand from the parental DNA. Thus, DNA replication is semi-conservative, right? Now, this was their hypothesis to begin with, that DNA replicates semi-conservatively. But at that time, there were other hypotheses of DNA replication that existed. Now, what were the three major ones? One was what we have seen has been experimented on and proven, that is DNA replicates semi-conservatively, right? What does that mean? We know that the parent strand both act as template for one new daughter strand, and now these two become separate DNA, like they are incorporated in separate DNA strands, right? Then there was one theory which said that there was a conservative kind of DNA replication that could occur, right? What would this mean? That uh, in during replication, these two strands would separate, right? They would act as template for two new strands, but at the end of replication, the two original parent strands would come and bind together, and the two newly synthesized strands would go and bind together, right? So this would mean that there would always be a heavy chain strand that would have been present if the same experimental setup would have been done on this also. So that experiment proved that it could not be conservative. Why? Because if it had been conservative, then by the end of the experiment, we would also have received the heavy chain band, right? That heavy only band, which was not available because it was semi-conservative. Then the third model was dispersive, right? Now, what does this dispersive model says? It says that DNA basically randomly, some part of the DNA, like it will randomly segregate, and after these random segregates are already present, they will denaturate, they will act as template for the new strands, then these segregated parent strands would merge together, and wherever the new strand they had been segregated from the original complex, there the new strand will be added, right? It was a very haphazard way of replicating, but through the same experiment, that is Michelson style experiment, how do we know that dispersive kind of replication does not occur? Because over here, as you can see, we just obtain three distinct bands, right? If there was dispersive mode of replication that could occur, we would receive many intermediate bands between these three distinct bands, right? For example, because there would be chains which had some heavy chain, few light chain, some would have more light chains, few heavy chain, right? It would not be 1:1 ratio for anything, right? So that is how we know that it is not even dispersive mode of replication that occurs, right? The same density gradient centrifugation has been shown over here also, wherein it is seen that originally, if we see, all of the bands that we receive are in the heavy chain bands. Then we receive intermediate bands, and with time, this intermediate and light band occurs, and as the time progresses, the intermediate band goes down, and the light band keeps increasing, because now they are only present in a media that is enriched with light chain of nitrogen, right? So this they continuously have a supply of, while uh, like to begin with, we only might have 100 of this N15 band E. coli, right? Now, among 1,000, those 100 will maybe just be 10%, right? So that is why they continuously keep on decreasing, while the other one keeps on increasing. So I hope that is clear. This brings us to our final question for the day, which says, during DNA replication, in which direction does the DNA polymerase 3 synthesize the new DNA strand? So is it in 5' to 3' direction, or 3' to 5' direction, or 3' to 3' direction, or 5' to 5' direction? Now, this is another thing that we have been studying for a while now. It is one of the very basics of this course, that DNA polymerase can only synthesize new strands in, especially polymerase 3, in the 5' to 3' direction, right? So that brings us to the end of the first segment of this lecture. I will move on to the second. I will actually stop sharing the screen, and then I'll move to the second segment. right? Just give me a very, very short break to have water. Okay, guys, I hope the first segment of this particular session was clear for you guys. Now we'll be moving to segment two, wherein, like last week, we discussed some other concepts in detail. We have a few concepts that we have prepared for this week as well. I'll share my screen for now. Yeah. Yeah, I hope my screen is visible to all. In the second segment, we will discuss some extra concepts that were not dealt with during this week's lectures. Right. In the discussion forum, I did not see many questions that needed to be addressed. Right. The major question was to upload PDF for ma'am's lectures. That unfortunately I cannot like do from my end. So if ma'am sees that particular request, I'm sure she'll do that. So that like neglects our discussion for the discussion forum doubts or problems. We'll just begin with the extra questions that we wanted to address, right? Or extra concepts that we wanted to address. The first one for this lecture being uh, a little bit of detail about DNA structure. Since we are discussing DNA in this know, high depth or detail, I would also like you to like remember a few points about its structure. I am sure you guys have studied that before. Just to remind that to you, because I feel that it should go side by side with the current knowledge about replication that we are gathering. So for the same, I have again used some material from Lodish that I did last week. Along with this, there is this very, very nice book. Like most of my molecular biology concepts, I have learned from this book, which is Molecular Biology of the Gene by Watson. Right, so I have included a lot of material from this book for this particular session, and we'll discuss that. Right. So, when we talk about structure of the DNA, right? So, beginning with the very basic, what is DNA made up of, right? We have already discussed its components. Deoxyribonucleic acid. So it has nucleotide bases. It has deoxyribose sugar, and it has phosphates that are attached to it, right? That those concepts we have dealt with. Let us talk about it in this structural as a structural entity, like we deal with when we are talking about it in case of chromosomes or in case of replication, etc. Right? So DNA is composed of polynucleotide chains, right? So polynucleotide chains, again, what does it mean? There's some background noise. Just a moment. Okay, I have just shut the door. I hope that helps a little with that noise that was coming. Okay. DNA is composed of polynucleotide chains, like we were talking about. What are these polynucleotide chains? It is basically um, um, uh, segment of nucleotides that is attached to one another continuously in a chain-like manner, right? As is what is represented over here. So there are multiple polynucleotides that are added. There is this A base that is added to one deoxyribose sugar and phosphate molecule that is in turn attached to this thymine that is again attached to guanine, right? Through this backbone that consists of the deoxyribose, the phosphate, etc. Right? So DNA is composed of polynucleotide chains, and the two chains of the double helix have complementary sequences, right? So we know what complementary sequences in with respect to DNA are. It basically means that A, whenever it is present, it will always have T as its partner, right? So we are saying that these chains are running anti-parallel. So its complementary partner will always be T, right? Or in case of RNA, when we talk about it, will be U, right? And whenever G or C is present, its consequent partner will be again C or G, right? So GC go together, they are complementary to one another. AT go together, they are complementary to one another, right? Then there is hydrogen bonding that is important for the specificity of the base pairing, right? So as you can see over here, there are hydrogen bonds that are present between these complementary bases that are important for the specificity of the base pairing. That is, if, for example, in front of T, while replicating over here, somebody comes in, like the DNA polymerase, uh, by default, it instead of A, if it brings a G, it will not be able to sit over here because this bond, the hydrogen bond, will be unable to be formed, right? And once that is unable to be formed, it will quickly know, the polymerase will quickly know that, okay, there is something wrong that I'm doing over here. And since hydrogen bond has not formed over here properly, uh, instantly there will be melting of whatever transient bond will also have been formed over here, such that this base will hang out, the incorrect base that was added, and through exonuclease activity, it will be corrected. Right, we'll talk about that also. Then when we talk about structure of DNA, we usually talk about handedness, right? So whether it is a left-handed structure or a right-handed structure. So on this, so I don't know how many of you are going to be willing to answer this in the first place, but just a moment. Yeah. But Okay, I've already written the answer. DNA is usually right-handed double helix, right? But why have I written usually over here? Why is it not always right-handed double helix? So before we talk about that, we'll talk about what this right-handedness and left-handedness is. So if you remember, when we used to study physics, we had some, oh, what was that called? I don't remember, right? We used to check some yeah, like when we used to study electromagnetic force, I don't remember that concept on the top of my head, but we used to like have this finger that, okay, this one is maybe force, this is magnetism, this is, um, okay, I don't remember it and I don't want to say that incorrectly. Okay, but I hope since you guys might have been closer to your 12th than I was, I hope you understand what I'm trying to say. So whenever you keep your right hand forward, for example, uh, your fingers will be facing a certain way, right? So if that is the way the DNA is looped, right? For example, it will begin over here and then it will go like this, right? Then it will be a right-handed helix. But if you keep your left hand forward and after that, when your fingers are facing the other way, if that is how your DNA is looped, it will be a left-handed helix, right? Mostly, as I told you, usually DNA is a right-handed double helix, right? But since I've added this word, that is usually, it is also possible that sometimes DNA is actually a left-handed helix. Now, what will determine this? Whether it is a right-handed helix or left-handed helix, right? What will determine this? So the sequence of bases or how they are present with respect to one another, the base pairs being present with respect to one another will determine whether it will form a right-handed helix or a left-handed helix, right? So in case of a right-handed helix, what happens is that usually if in any random fashion the DNA bases are present, they will form a right-handed helix, right? And some forms of DNA in which right-handed helix are present are the B form, A form, etc., right? But if the DNA contains alternative purine and pyrimidine residues, it can fold into a left-handed as well as right-handed helix, right? So what can happen if there is alternating purine pyrimidine residue that is present, it can also sometimes form a left-handed helix, right? So only examples of these kinds of helices are a Z type of DNA, right? So I'm sure you have gone through these different types of DNA structures. One is the most commonly found one is B. Then sometimes A kind of DNA is also found. Sometimes Z kind of DNA is also found. I'm sure you have gone through that table that represent how they are different from one another. This is one of the differences, right? So if we talk about why or how can this happen, that sometimes it will form a right hand helix, sometimes DNA can form a left hand helix. How would this phenomena happen, right? For that, there are glycosidic bonds that are present, right? So what are these glycosidic bonds? These are the bonds that are present between the bases and the ribose sugars. Okay, when these glycosidic bonds are present, they could be in two conformations. One is a syn conformation, wherein it will look like this. Once we talk about adenine, this is the adenosine that is present, and this is how syn adenosine will look like. But the other form is anti-adenosine. If it is present in this anti-adenosine form, this is how it will look like, right? So in right-handed DNA, it is always in the anti-conformation. Okay, once we are talking about the most common form of DNA, that is the right-handed DNA, it will always be in a anti-conformation, and that anti-conformation we now know favors right-handed helix. But in left-handed helix, what happens is that the fundamental repeating unit is usually a purine pyrimidine dinucleotide. So as we said, there is an alternate purines, pyrimidines, right? And as the dinucleotides, they are present with glycosidic bonds in anti-conformation at pyrimidine residues and syn conformation at purine residues. So at A and G, they will be at this syn conformation like this, and at T and C, they will be at anti-conformation like this. Right, so once they are present in an alternating unit and one has a syn conformation, the other has a anti-conformation, what will occur? It will kind of form a zigzag-like structure, right? So the change to syn position at purine residues to alternating anti-syn conformation gives the backbone of the left-handed DNA a zigzag-like look, right? For example, which is like it is going like this, then this, then this, then this. Uh, it might be a little difficult to see in that illustration, but it will actually look like this, right? One is going in an anti-conformation out towards outside, the other is going in a syn conformation towards inside, then again anti, then again syn. So it will actually look like a zigzag, right? And consequently, now this zigzag will uh, be turned also, right? Like how a DNA is turned. So instead of the straight kind of DNA that we see now, it will be a zigzag kind of DNA, right? So this is basically what is known as ZDNA. Coming back to the general structure of DNA, it also has a double helix. The double helix has two grooves, right? One is a minor groove and one is a major groove. So I hope you all are aware of what the minor groove and major groove are. So as the name suggests, what is going to be a major groove? It is going to be the larger groove, and minor groove is going to be the smaller groove, right? What are these grooves? It is the spaces that are kind of present towards the outside of the bases, right, of the phosphate backbone. For example, if we talk about DNA, we know this is how we kind of draw our DNA, right? We will never, although there is that A kind of DNA that has all, sorry, that almost has equal major and minor grooves. Why does it have equal major and minor grooves? Please ignore these spikes that are going off. Because A kind of DNA kind of looks like this, right? So over here, this groove, they kind of look similar because the angles that they are presented are similar, very, very similar. But the most common kind of DNA that we talk about is B form of DNA, like the one that has been drawn over there. Like, sorry, once we draw this kind of DNA, how do we draw it? We draw it like this. Okay, I have been unsuccessful in drawing it, but okay. So we just look at what is already drawn over here, right? So over here we see sometimes there is a big gap or a big groove, and sometimes there is a small groove or a small gap, right? And they are opposite to one another. With one big gap, it will have a small gap on the opposite. With one small gap, it will have a big gap on the opposite, right? So this is a major groove and this is a minor groove. Okay. So we know that two different kinds of grooves occur in case of double helix DNA structures. Now, why does that happen? Before that, let us just read the statement. The major groove occurs where the backbones are far apart, as we can see over here, right? So this backbone is farther from this backbone as compared to this and this backbone, right? And the minor groove occurs where they are close together, right? As we have already seen over here. Now, the major groove is rich in chemical information. This basically answers the question of why, in the first place, major and minor grooves occur, right? So it will become clearer to you once we are done with this discussion. At least I hope that. So what happens once there is this major groove and minor groove that are present? They are the major grooves are rich in chemical information. I will tell you what I mean by this chemical information, right? So as I was saying, uh, we have said over here that the major groove is rich in chemical information. Now, what is the nature of this chemical information? What does it do? We will come to that. Before that, let us talk about some more properties about this major and minor groove. So one more way to distinguish it completely is that the major groove has a groove angle of about 240 degrees between two different nucleotide bases, right? So on this end, if you see, there is an angle of about 240 degrees that is present when it is a major groove. But in case of minor groove, this groove angle reduces to about 120 degree, right? So that is another kind of a definition that we can give for a major groove versus a minor groove, right? Now, along with this, on the right hand side, I hope you can appreciate this image that I have, or this illustration that I have kept, right? So this has been picked from Nucleic Acid Research, which is uh, one of a very good journals. Uh, in this illustration, what the authors have represented is that in the minor groove, there are some chemical signals that are there, right? Now, what are these chemical signals over here? The red dot represents hydrogen bond acceptor, blue dot represents hydrogen bond donor, yellow represents methyl group, and the uncolored or the white colored dot represents non-polar hydrogen, right? So, and they have all been given a single letter denotation, notation also, that is A, D, M, and N. We will come to that later, and for that, please remember this key, or please understand this key, right? So over here, we can see that when we talk about major group, in case of G and C, when these two are present, what happens in case of major group? There is a particular signal or signature that is present, right? So this particular major group, when G is bonding with C, in this particular orientation, where G is on the left and C is on the right, or G is on the positive strand and C is on the negative strand, then what will happen? It will have a specific kind of signature. What is this signature? Let us read it. That first there is a hydrogen bond acceptor, then again there is another hydrogen bond acceptor, then there's a hydrogen bond donor, and then there is a non-polar hydrogen, right? If we write it as the denotations that are given, or the notations that are given to it, it will be AADN, right? This G to C linkage will be read as AADN, right? Similarly, when A is bonded to T, the major group again has a particular kind of signature. What is the signature? Hydrogen acceptor, then hydrogen bond donor, hydrogen bond acceptor, and methyl group. That is ADAM, right? So this is the chemical information that we are actually talking about, that this major groove is holding, right? So as we talked about, if A to T are present or AT base pairing is there, then ADAM, like we discussed, is the denotation or signature that is there. DH is the signature that is present when G is bonded to C, right? That also we saw. If it is inverse, if T is present on this strand and A is present on this other strand, then it will have a different signature, right? It will have MADA as the signature that is present on the major groove. On the contrary, if C is present on this strand and G is present on this opposite strand, it will again have a different signature, which is HDAA, right? So it is again a very different signature that is present over here, right? So what does this mean? Over here, instead of N, I have used H, right? So H over here means non-polar hydrogen. It is not following the key that is given over here, but this is actually the more followed key, right? This is something I picked up from Watson, while this particular illustration I picked up from this paper in NAR. Okay. So these patterns are important because they allow proteins to unambiguously recognize DNA sequences without having to open and thereby disrupt the double helix. So how do we say that major groove is rich in chemical information? Because if such kind of signatures exist, right, without having to open this double helix from outside only through seeing these signatures, there will be some of the proteins that will understand, okay, this particular region has AT bond, maybe I have to go and attack this particular region. This particular region has a GC bond, maybe I have to go and attack this particular region, and so on, right? Maybe that is how uh, the melting starts from an AT-rich region. Maybe that is how an AT-rich region is identified in the first place, right? So that is why we are saying that this particular major groove, because of its definite signature that is present, is rich in chemical information, right? Now the question arises as to why we don't say the same for the minor groove. Yeah, right? Because over here also we can see that there are some electron pairs that are present, which in this case in G and C, it will represent hydrogen bond acceptor A, then hydrogen bond donor D, and then again hydrogen bond acceptor A, right? So it will be ADA, right? In case of G and C. But again, if C is present over here and G is present on the other strand, this signature will again be the same, ADA, right? And also because it is a kind of signature that is read the same from up and down, we will not know from which way we are going or proceeding, right? The same is the case with A and T. In case of minor groove, they have a signature that is AHA, right? So this AHA will be same for AT as well as TA. So in case of minor groove also, if we see, there could be some information that could be gathered that whether AT is present over here or GC is present over here, right? We can't determine whether it is GC or CG or whether it is AT or TA, right? That can't be determined. And since they are palindromic, right, these signatures are palindromic, that is, they are same when read from the forward and the backward, we will not be able to understand like which one is the correct like direction, right? So that is why we say that minor groove is not that rich in chemical information as is major groove, right? So I hope this concept was clear. This was one of the major things I wanted to actually get across. And since that is over, maybe we can move on. Next, we continue to talk about the structure of DNA. The point that we now will speak upon is double helix exists in multiple conformations. So these are conformations that we have been speaking about for a while now. This is actually how I was trying to draw that ZDNA also. So this you can see, maybe I will zoom it for you. This is how it looks, right? So if it is a kind of DNA, as you can see, the major and minor groove does not have much difference between them, right? Between the size, there's not much difference between the size of these major and minor grooves. While in case of B DNA, there is a significant difference between major groove, which is very large, and minor groove, which is considerably small over here, right? Then there is ZDNA that is present over here, which again has almost equivalent major and minor grooves, but over here, okay, major and minor are not equivalent because if you see over here on the right side, this is the major groove, which is considerably bigger than the minor groove, right? The only difference over here is the zigzag kind of alternating structure that is present, and along with that, instead of uh, like it being a right-handed helix like the other two, A and B, it is a left-handed helix, right? ZDNA. So these are some of the conformations in which DNA double helix can exist. This is one of the tables. It is a very, not a very information intensive one. There are much better tables that you can get. This is just an example. For example, if we talk about A, B versus Z, the shape and about the shape of these DNAs, if we compare that, then B has an intermediate shape. Z DNA, when we look at it, is the narrowest among all, and aDNA is the broadest among all, right? Because it has those loops that are going like, and it is similar on both the ends. So it has to maintain that end-to-end shape. So it is broadest. Then if we talk about rise per base pair, right? That how in how many base pairs there is a rise over here, right? So it is also the highest in case of Z, as you can see over here also. So between the distance is the highest over here, that is 3.8 angstrom. Then it is for B, that is 3.4 angstrom, and then comes A with 2.3 angstrom between for the rise per base pair. Then the helix diameter for A is around 26 angstrom. For B, it is around 20 angstrom, and for Z, it is around 18 angstrom. Right? That is what we are basing this broadest, intermediate, and narrowest on. If we talk about the screw sense, then A and B are both are both right-handed, while Z is left-handed. If we talk about glycosidic bonds, then A and B only have anti kind of glycosidic bonds, but because Z has both alternating anti and syn kind of glycosidic bonds, that is why it has this zigzag-like structure, and that is why it is also left-handed, right? Then base pairs per turn of helix. So how many number of base pairs are there per turn? It is 11 in A, 10.4 or about 10 in B, and 12 in Z, right? Then pitch per turn of helix, that is a height that is there per turn of helix, is the least in case of A, that is 25.3 angstrom, because it is broader, right? So it is second highest for B with 35.4 angstrom, and it is the maximum pitch is present in case of Z form of DNA, that is 45.6 angstrom, right? Then tilt of base pairs per base pairs from perpendicular to helix axis has 19 degrees tilt in case of this A form of DNA, 1 degree only in case of B form of DNA, and 9 degrees in form of Z form of DNA, right? So that was it for the conformation, the various conformations of DNA. And one last thing that we want to speak about the structure of DNA is that it is possible for DNA strands to separate and to reassociate. That is to denaturate and then to renaturate, right? Now, how does it happen? Most commonly, it is due to the presence of heat or other enzymes that are present, right? So if there is a native double-stranded helix that is present, which is a DNA, if heat is provided to it, or in case of some enzymes that are present, it could partially unwind, right? If in case of heat, if we are talking, if more heat is given, then it will completely denature and its strands will dissociate from one another. But if it is cooled and it is brought back to an optimal temperature, they will again reassociate, and renaturation of this double-stranded helix would occur, right? So a recap for the structure of DNA for whatever we have discussed up till now in the second segment of the lecture is that DNA is composed of polynucleotide chains, right? The the two chains of double helix have complementary sequences to one another. The hydrogen bonding is important for the specificity of base pairing. Then, DNA is usually a right-handed double helix. It can sometimes also form a left-handed helix. Then the double helix has minor and major grooves, and the major groove is rich in chemical information. The double helix exists in multiple conformations, and the DNA strands can separate and reassociate, right? With change in optimal temperature, that is, with the help of some enzymes that are present. So I would yearn you to go through this recap once while I take a short. Yeah, so I also wanted to just make sure that I had sufficient charging, so I plugged in a charger. I'll just stop sharing and share again so that there's no issue with the current sharing, right? So I'm sharing my screen again. Okay. Yeah. So what we talked about last was the structure of DNA, and we did a short recap on whatever we had gone through. Then now we are going to talk about DNA synthesis, right? So what is DNA synthesis? It is basically one of the prerequisites to replication of DNA, right? So first it or replication, sorry, a prerequisite to replication of cells, right? So DNA needs to be synthesized so that now we have enough DNA to give to two different cells that are going to be formed, right? So what does DNA synthesis require, right? It requires deoxynucleotide triphosphates and a primer template junction. So these are the two prerequisites for the synthesis of DNA. One is deoxyribonucleotides, uh, uh, and deoxynucleoside triphosphates, right, which have three phosphates that are attached to a deoxyribose sugar, which in turn is attached to a base, which is one of these, that is A, C, G, or T, right? Along with that, since even the DNA polymerase, although it can add this DNTPs to a growing nucleotide chain, these deoxyribonucleotides to a growing chain, right? That is how it can elongate the DNA, but it cannot start the de novo synthesis of DNA chain, right? For that, we need a pre-made primer, right? So there needs to be a primer that is made. Now this primer is going to go and sit on the template to this already made primer, which already has one 3' free end. Later, the DNA polymerase can act and it can keep on adding DNTPs to this already present or existing chain, right? That is how it works. So, when we talk about DNA synthesis, we also say that it is synthesized by extending the 3' end of the primer. Now, this is one fact that we discussed while we were also solving the questions for previous year, right, for this particular week. So as you can see over here, that the DNA polymerase will always extend or elongate in a 5' to 3' direction. That being said, there will always be a 3' at the 3' end of the phosphate backbone. There will always be a hydroxyl group that will be reactive. It will go and it will basically react to the incoming phosphate group of this particular DNTP that is being added, right? Once this reaction happens, a pyrophosphate will be removed by pyrophosphatase, right? By it will be removed, and through pyrophosphatase it will be then degraded down to two phosphates, and this single phosphate contained deoxyribose sugar and amino acid will come and it will join this chain, right? So that is how DNA will eventually be synthesized. Now, the hydrolysis of pyrophosphatase is actually the driving force for DNA synthesis. Now, this could be one of the questions that is asked to you. What is the driving force for DNA synthesis? It is hydrolysis of this pyrophosphate, right? So, if we talk about any phosphate to speak of, over here, X represents any of the phosphates, for example, A or G or T or C, right? So, X over here could either be A or it could be T or it could be G or it could be C. So since I have not written it in a key over here, I hope it is very, very clear to you guys. But despite that, I have written it for now. So this XTP could, if we talk in context of adenine, right, or adenosine, we will say that ATP plus AMP raised to power n, right, into power n would basically give, uh, what is this? It is the existing polynucleotide that is present, right, to which a new DNTP is being added, right? So it will basically form this particular XMP, which has a monophosphate. The polynucleotide that is present has a monophosphate backbone, right? So it will like add one nucleotide to it, and it will form a monophosphate backbone. But from this XTP, which had three phosphates, two phosphates will be released, right? These two phosphates are the pyrophosphates, right? Once this reaction happens, it will release free energy, which will be equivalent to 3.5 kilocalorie per mole, right? After this, what will happen is that these two phosphates that are joined together, pyrophosphate, by the action of pyrophosphatase, what will happen is that they will also break down into two individual phosphates. And this again will release a free energy which is equivalent to 3.5 kilocalorie per mole, right? And after that, the overall reaction, if we look at it, there is an existing polynucleotide chain to which a trinucleotide is being added, right? This trinucleotide will be added, but two of its phosphate will be lost, or the as pyrophosphate, which later converts into two single phosphates. And this entire process will release 3.5 plus 3.5, that is 7 kilocalorie per mole of free energy, right? That is a lot of free energy, and it is the driving force for DNA synthesis, right? So a small recap for DNA synthesis would be that DNA synthesis requires deoxynucleoside triphosphates and a primer template junction, right? DNA is synthesized by extending the 3' end of the primer, and hydrolysis of the pyrophosphate is the driving force for DNA synthesis. So again, I hope that is also clear. Then this is again a recap from what we have already studied in our lecture series that was given by ma'am, right? So what are we looking at? It is a very, very complex diagram if we look at all of it at once. But if we break it into parts and look at it, like if we break it into the parts that it has already been assigned as, right? The numbers that are present, 1, 2, 3, 4, 5, right? We will look at it in that order only. So what are we looking at? We are looking at a replication fork. Now, which region over here signifies a replication fork? This particular region, right? I don't know whether okay, I will just zoom out, because if I zoom in, I cannot use my pointer, right? So this particular region is the replication fork, right? Because this is a existing DNA, which is represented by dark blue and light blue. Now, this is being broken apart into a replication fork such that both of the light blue is acting as a template for this dark green, and the dark blue is acting as a template for this what red colored new DNA that is being synthesized, right? So at this replication fork, we see that there are multiple other components that are also present, right? Let me first clarify. We are looking at a replication fork of SV40 DNA. SV40 is a viral particle, is a DNA virus, right? It is a form of DNA virus. I'm not recalling what exactly SV stands for, but it is a virus and it has DNA as its genetic material. So it is a DNA virus, right? So we are looking at its replication fork. It is a eukaryotic DNA replication fork. So as we talked about, eukaryotes have three major polymerases that are present, right? A polymerase alpha, polymerase delta, and polymerase epsilon. So as we see, polymerase epsilon over here is continually going, and it is synthesizing DNA on the leading strand. This one, on the other hand, the one that has RPA that is bound to it, and a loop that is continuously being formed, which is known as a trombone structure, is signifying the lagging strand, right? It is also written over here. It is a lagging strand. This one was a leading strand. So in the lagging strand, the main DNA polymerase is the polymerase delta, right? Along with that polymerase alpha is a primase for this strand, right? It will go and it will add primers such that on that DNA polymerase delta can act, and it can complete the synthesis in between this primer and the next primer, right? Along with that, the other components that we are seeing over here are, one is a large T antigen. What is this large T antigen? It is a helicase. What will a helicase do? A helicase has a major role to play in formation of this replication fork, because it will denature the existing DNA, right? So this is the helicase that has been enlarged over here in B also, right? After this, going to 2 is RPA. Now, what is RPA? RPA is basically a protein that is going to bind to these are individual RPA proteins, right? These are going to go and bind to single-stranded DNA, right? Until and unless a new strand is being synthesized on it, they are going to go and sit on the single-stranded DNA so that it doesn't uh, come and it binds to itself. There is no self binding that occurs. So it is going to stick on those bands such that they are drawn apart, right? So that is what this RPA is. Then coming to three is this PCNA. PCNA, as ma'am talked of extensively, is a clamp, right? Over here, this clamp will be present in front of the polymerase, right? So it is present in front of all polymerases. Over here in polymerase epsilon, we can see it near polymerase alpha, we can see it, then near polymerase delta, we can see it. So what will this clamp do? It will basically hold the polymerase onto this particular DNA that it has to remain on, right? So it will go and it will bind, and this PCNA, this open loop, will close around the DNA. It will close such that now this polymerase, until its work is done, it cannot fall off this particular DNA that it has to work on, right? So that is PCNA. It will go and it will form a loop around double-stranded DNA. Then after PCNA, we have polymerase alpha primase, right? This I have discussed that it is the third polymerase that is present over here. It is going to add primers to the lagging strand. Then after this, fifth, we have polymerase delta. What is this polymerase delta going to do? This polymerase delta is basically going to help on lagging strand DNA synthesis, right? Apart from this, one segment that we did not discuss was this RFC PCNA. Since PCNA is the clamp, RFC over here is a clamp loader, right? You can see that when RFC is present, PCNA is not completely circular, right? So it is still open so that it can go and it can sit on that DNA where it has to remain. Right, after it will sit properly, it will close this particular structure also, and the clamp loader will then go away because loading of the clamp has been done. Right, so this was just recollection of what we have already studied during this week. Yeah, so uh, again, it is the same DNA virus that we are talking about. So over here we talked about DNA virus SV40, right, and its replication form. Now these images over here on the left are actual electron micrograph images of this SV40 viral DNA being replicated, right? So you can see that there was a replication fork that arised, and it is a bidirectional replication that is taking place, that is on both sides of the replication bubble. It will keep extending, right? And you can see in the third electron micrograph that it has extended to good enough amount, right? So that is how the DNA replication will occur over here. Now, having said that, this virus actually, although it is a DNA virus, it has circular DNA, right? So the DNA is not linear generally, it is a circular DNA, but it has restriction sites like most viruses, right? So on its EcoR1 restriction site, EcoR1 goes and acts. It is a restriction enzyme, it will go and act, and it will basically convert this circular DNA to linear DNA. Once it is converted to a linear DNA, now its replication will proceed like any linear DNA replication would have proceeded, right? So when we talk about replication being bidirectional, what do we mean? That if there is a replication bubble that is being formed, right, like this, or rather a better representation would be that there is a replication bubble, and on either side of it, it is still not unbound, right? Something like that. In this replication bubble, say that the origin will be in the middle, right? Say this first strand. Now let us talk about in this particular example, because that is how I will be able to tell you that, okay, this is the strand we are talking about. Say this first strand is in 5' to 3' direction, and this other one is anti-parallel and it is in 3' to 5' direction, right? Now, how can polymerase always act on? In which direction? It will always act in 5' to 3' direction, right? So it will synthesize a new chain in 5' to 3' direction. So that means that in this particular strand, the top strand, this will be the leading strand in 5' to 3' direction, and the other one will be the lagging strand, right? Consequently, in the other one, this will be the leading strand and this will be the lagging strand, right? That is how this will continue. Like once this much has been done, then the replication bubble will further open up, right? It will open up more. Once it has opened up more, this will continue. The leading strand synthesis will continue. And the lagging strand synthesis will also continue like this, right? Until this entire DNA has opened up. And from here to here, there is one leading strand. From here to here, there was another leading strand, right? And then over here, there was lagging strand that was present. Over here, there was lagging strand that was present. And now DNA1 will come and it will fill in these gaps that are present, and then ligase will come that will completely seal the gaps that were present, and now there will be two DNA that will now be formed, right? One will be this top DNA and the other will be this bottom DNA. So I hope that is clear. Then we talk about proofreading by DNA polymerase, right? So DNA polymerase, what is one of its function? One of its function is to go and synthesize new DNA. But another of its function is proofreading, right? It has to synthesize DNA, but that DNA also needs to be accurately synthesized, like we talked about, right? It should not have any incorrect bases that are present, because that will simply lead to mutations being formed, right? So what will DNA polymerase do? Or rather, let us talk about what does DNA polymerase first look like, right? So all DNA polymerases have a similar three-dimensional structure which resembles half open right hand, right? So like the one that I have represented over here. So this was, okay, it didn't get zoomed in. This was another of the illustrations that I picked in from this particular one on the right hand corner, is the one that I picked in from Watson again. So it shows, you can see that it kind of looks like fingers. When you put your right hand forward, then there is a thumb, there is fingers, there is palm, right? And each of these have different jobs to do, right? But that is how DNA polymerase kind of looks like. And what is its job? Its job is to kind of go and grip the template primer between its palm, so that after that it can start the synthesis of the new DNA segment in front of this primer that was already present, right? And taking the signal from the template. So the fingers bind the single stranded segment. As you can see over here, this is another kind of representation over here. This like we were talking about hand like this. Now we are talking about it in this orientation, right? So if I talk about hand in this orientation and I open it like this, like, right? So this lot will be fingers, this lot will be thumb, and this will be the palm. That is how this representation is being made, right? So this in this representation, we can see these fingers which have this polymerase activity, right? So this finger region will have a polymerase like activity. This polymerase activity will be the same 5' to 3' adding polymerase activity. The thumb has a different role that we are not talking about right now. And this end of the palm, right? This end of the palm will have a exonuclease activity in case something goes wrong. When we are actually synthesizing new DNA, it will come back over here. How will it come back? Let us read, right? The fingers bind the single stranded segment of the template strands. So the as you can see over here, and the polymerase catalytic activity, or POL, lies in the junction between the fingers and the palm. So it lies over here, right? As long as the correct nucleotides are added to the 3' end of the growing strand, it remains in the polymerase site. So as long as it is going correctly, everything is being added in a perfect way, it will remain in this particular site only. But what happens sometimes, if there is an incorporation of an incorrect base at 3' end, it causes melting of the newly formed end of duplex, right? So how will this melting occur? I told you when we were talking about hydrogen bond formation, that since an incorrect base was being added over here, it will not correctly be able to form a hydrogen bond with its with what was already present on the template strand, because it is probably not complementary to that particular base pair, right? So the melting will occur over here, and once melting occurs, it will leave this site, the polymerase site, and it will now come to this exonuclease activity site, right? As a result, the polymerase pauses and the 3' end of the growing strand is transferred to the 3' to 5' exonuclease site. Right. So now what is this exonuclease site? It is 3' to 5'. So it will remove nucleotides in 3' to 5' direction. The other one was adding in 5' to 3' direction. It is removing from 3' to 5' direction. Right. This site is about 3 nanometers away, and the mispaired base and probably other bases are also removed, right? Then subsequently, after the removal of this mismatched base has occurred in this exonuclease site, the 3' end will flip back to the polymerase side and the elongation will resume. Right, so it will flip back. That is why this reversible arrow, and then elongation will resume again. Right, so I hope that uh, short but important concept was clear. Then I have mentioned or put in this table that represents the characteristics of various DNA polymerases. So as to speak, the eukaryotes can have all of them have 5' to 3' polymerase activity. That is why they are characterized as polymerases in the first place. While a few of them can also have 3' to 5' exonuclease activity, like the one that we were describing. So that was being talked about this DNA pol 3, right? Even DNA pol 2 and DNA pol 1 have this 3' to 5' exonuclease activity. Then there is one kind of exonuclease activity, that's this 5' to 3' exonuclease activity, right? That is present only with DNA pol 1. So why is this important? Because how is the synthesis occurring? It is occurring in 5' to 3' direction. Now, in the 5' region, the first few sets are not the DNTPs, right? It is a primer. Primer needs to be removed, and in place of that, DNTPs need to be added. So now we can't go on from this 3' to 5', removing every nucleotide to remove that primer, right? There needs to be one kind of polymerase that can directly come and act on 5' side and start removing from 5' direction, right? So that is what this DNA polymerase 1 does. So thus the function of DNA polymerase 1 is to remove and replace primers. The function of DNA polymerase 2 is DNA repair. It restarts replication after damaged DNA halt synthesis. DNA poly 3 is used to elongate DNA. DNA poly 4 helps in repair of DNA, and DNA poly 5 also helps in repair of DNA and in translesion DNA synthesis, right? So sometimes it helps you overlook some lesions that are present in DNA and continue the synthesis so that that halt is not there for long. This also has been discussed in ma'am's lectures, translesion DNA synthesis. I hope you remember that. So that brings us to the end of this segment also. That was it from my end for today. I hope uh, it was of some benefit to you guys. And if you have any questions, we can go ahead and we can deal with those questions. If not, I'll end the session. Okay, so no questions. That is what I'll assume. Okay, guys. So in that case, thank you so much for being a part of the session, for listening so patiently, also being involved with the question answers, answering like from time to time. So I did not have to prompt a lot. There were some answers that were coming along the way. Uh, that is really nice. I hope this session benefited you. I will just stop the recording. Yeah.